Hey everyone, I am Dr. Maryam. Welcome to chapter 25 Heredity and Hereditary Diseases. This chapter covers how traits are passed from parents to children, from basic genetics to complex inheritance pattern, and the disorders that can result from mutation. We will talk about genes, chromosome, alleles, dominance, meiosis, Punnett squares, sex determination, sex-linked traits, mutation, and specific genetic diseases. We will also cover genetic counseling and prenatal screening. Let's get started. Genes and chromosomes. A gene is a segment of DNA that codes for a specific protein, like instructions for eye color or blood type. Different versions of the same gene are called alleles. For example, gene for eye color has alleles for blue, brown, green. Humans have 46 chromosomes, 22 pairs of autosome, non-sex chromosome, and one pair of sex chromosome, XX for female, XY for male. And this is the image of the chromosome. Let's discuss the image, this specific image in detail on the next slide. Genes and alleles. For each gene, you have two alleles, one from each parent. If both alleles are the same, you are homozygous. If they are different, you are heterozygous. A dominant allele only needs to be inherited from one parent to show its effect. A recessive allele must be inherited from both parents to be expressed. So a person can carry a recessive allele without showing the trait. They are a carrier. Phenotype and genotype. Phenotype is what you can see or measure, like brown eyes or type A blood group. Genotype is the actual genetic makeup. For example, a person with the genotype BB for dominant brown eye gene has brown eyes, phenotype brown, but they carry a recessive blue eye allele. That's important for inheritance. Meiosis. Gametes, sperm and eggs are produced by meiosis. It involves two divisions. The first meiotic division separates the two homologous chromosomes, one from each parent. The second meiotic division separates the two identical sister chromatids, so one diploid cell 46 chromosomes become four haploid cells 23 chromosomes each. The slide asks, how many cells are produced in one complete meiosis? The answer is 4. Starting with one diploid cell, after two division you get 4 haploid gametes. Each has 23 chromosomes, so the answer is 4 cells. Punnett square. A Punnett square is a simple grid that shows all possible combination of alleles from a parental cross. We use a capital letter for the dominant allele like B for brown eyes and lowercase letter for recessive allele, lowercase b for blue eyes. The parents' alleles are placed on the top and side, and the boxes show the possible genotypes of offspring. Let's say we cross two heterozygotes BB into BB. The Punnett square gives BB 25%, the uppercase lowercase b 50%, both lowercase b 25%. The recessive phenotype appears only in lowercase b, that's 25%. The heterozygous uppercase b lowercase b percentage is 50. These are classic Mendelian ratios. Sex determination. Sex is determined by the sex chromosome. Female have two X chromosomes, male have one X and one Y. The Y chromosome carries the SRY gene that triggers male development. The X is much larger and carries many genes unrelated to sex. That's why sex-linked traits are usually on the X. The diagram shows how sex is inherited. The mother always contributes an X, the father contributes either X making a girl or Y making a boy. So the father determines the baby sex. Sex-linked traits. Sex-linked traits are carried on the sex chromosome, mostly on the X. Because males have only one X, any recessive allele on that X will be expressed, even if it is recessive. That's why color blindness and hemophilia are much more common in males. Female can be carriers, they have one normal X and one affected X, but they don't show the disease because the normal X dominates. A carrier female for an X-linked recessive disorder has one normal allele and one recessive allele. For example, if normal is XH for hemophilia and XH a carrier, so the X dominant and the X recessive, so she doesn't have the disease but can pass the affected X to her children. Pop quiz. What is the term for all the variants of particular gene? A. Chromosome. B. Ribosome. C. Nucleotides. D. Alleles. I will pause for a few seconds. Think about it. And the answer is D. Alleles. What are the variants of a gene called? That's allele. Chromosomes hold DNA. Ribosomes make proteins. Nucleotides are DNA building blocks. So the answer is definitely D. Alleles. Traits are determined by both genetics and environment. Single gene inheritance is rare, like cystic fibrosis. Multifactorial inheritance is much more common. Traits like height, skin color, and risk for heart disease involve many genes plus environment. Even a purely genetic trait can be influenced by environment. For example, diet affects the severity of phenylketonuria. Gene expression. Just having a gene doesn't mean it's always active. Gene expression can be influenced by age, sex, like baldness genes require male hormones, other genes, epistasis, and environment, temperature in Siamese cats affect fur color. So genotype doesn't always equal phenotype. Variants of simple dominance. Not all inheritance is simple dominance slash recessive. Codominance, both alleles are expressed. Example, blood type AB. You have both A and B antigens. Incomplete dominance, the heterozygous shows a mix or intermediate phenotype. Example, sickle cell trait. Heterozygous have normal hemoglobin but have been sickle cells under low oxygen. They are resistant to malaria, which is why the allele persists. Genetic mutation. A mutation is a change in the DNA sequence. It can appear from chromosome breakage, copying errors during cell division, or abnormal genetic exchange like crossing over mistakes. Some mutations are spontaneous, others are induced by mutagens, radiation, chemicals, certain viruses. Not all mutations cause disease. Some are neutral, some even beneficial. The slide shows crossing over during meiosis. It's normal, but if it goes wrong, it can cause deletions, duplications, or translocation. Mutations can be gain of function. The protein is overactive or has a new activity. Example, cancer from mutated growth factor receptors. These tend to be dominant. Loss of function, the protein is missing or doesn't work. Example, sickle cell anemia. These tend to be recessive because one normal copy can often do the job. Mitochondrial inheritance. Mitochondrial DNA is a separate from nuclear DNA. It's passed only from the mother to all her children. Sperm doesn't contribute mitochondria. Mutations in mitochondrial DNA causes diseases like Leber's, hereditary optic neuropathy. Inheritance is maternal. An affected mother will pass it to all her children, but an affected father will not pass it. Congenital means present at birth. It can be genetic or not. For example, fetal alcohol syndrome is congenital but not hereditary. Hereditary means transmitted through genes, but the disease may not appear until later in life, like Huntington disease. So, not all congenital conditions are hereditary, and not all hereditary conditions are congenital. Causes of congenital disorders. Congenital disorders can be caused by maternal infections like German measles, which is also called rubella, teratogens, talidone, certain medication, ionizing radiations, alcohol, fetal alcohol syndrome, cigarette smoking, low birth weight, poor nutrition, folic acid deficiency causes spina bifida, and many causes are idiopathic unknown cause. This child shows the facial features of fetal alcohol syndrome: smooth philtrum, thin upper lip, small eye openings, and intellectual disability. Alcohol is a potent teratogen. There is no safe amount during pregnancy. Spina bifida is a neural tube defect where the spine doesn't close properly. It can be prevented with adequate folic acid before conception and during early pregnancy. This image shows the spinal cord exposed. Genetic disorders. Chromosome number disorders. Down syndrome, trisomy 21, three copies of chromosome 21. Klinefelter syndrome, XXY, male with extra X, tall, small testes, infertility. Turner syndrome, XO, female missing one X, short, web neck, underdeveloped ovaries. They are usually not inherited. They result from non-disjunction during meiosis. Autosomal dominant disorders need only one copy of the mutated gene. Huntington disease, progressive neurological deterioration symptoms appear in the middle age. Marfan syndrome, connective tissue disorder. Patients are tall with long limbs, lens dislocation, and risk of aortic dissection. Each child of an affected parent has a 50% chance of inheriting it. And these are the images of Down syndrome, Klinefelter syndrome, and Turner syndrome, which we just discussed the changes in the chromosome numbers in the previous slide. Recessive genetic diseases. These are the examples of autosomal recessive disorders. PKU. Inability to metabolize phenylalanine, causing brain damage if diet not controlled. Sickle cell anemia. Abnormal hemoglobin, RBCs, sickle under low oxygen. Cystic fibrosis, thick mucus in the lungs and pancreas. Tay-Sachs, lipid accumulation in neurons, fatal in childhood. Fragile X, intellectual disability. Osteogenesis imperfecta, brittle bones. Neurofibromatosis, nerve tumors, caries don't show the disease. Genetic counseling. Genetic counseling is for couples at risk of having a child with a genetic disorder. Indications: mother age over 35, family history of genetic disorders, fertility treatment, IVF, or abnormal prenatal screening. The counselor takes a detailed family history, draws a pedigree chart, and may order lab tests, karyotyping, DNA analysis. The couple then makes informed decisions. This is a classic pedigree question. The answer depends on the inheritance pattern. If the trait is recessive, the normal children could be either homozygous dominant or heterozygous carriers. Without more information, the possible genotypes are uppercase A or uppercase A and lowercase a. They cannot be lowercase a because they are normal. Prenatal screening includes first trimester test, knuckle translucency ultrasound, measure fluid at the back of the fetal neck, pregnancy associated plasma protein A, and HCG. Second trimester is the quadruple screen alpha fetoprotein AFP, estriol inhibin, and HCG. Abnormal results may lead to diagnostic test, amniocentesis, needle into amniotic sac, cells are cultured, or chorionic villus sampling, which is taken from the placental tissue. These allow karyotyping to see the chromosomes. This shows an amniocentesis procedure, ultrasound guided needle into the amniotic cavity to collect fluid containing fetal cells. Karyotype. Look at this karyotype. Count the pairs. There are 23 pairs, so 46 total. The sex chromosomes are XX, so female. Now look at chromosome 21. There are 3 copies, not 2. That's trisomy 21, which is a Down syndrome. So the answer to the question you can see on the slide. Look closely at this karyotype. How many chromosomes are present? What is the gender of the baby and what genetic disorder is represented? The genetic disorder here is Down syndrome because of the trisomy 21, 3 chromosome 8 number 21. Treatment for genetic diseases depends on the condition. Dietary control for phenylketonuria, no phenylalanine diet, maple syrup urine disease restricted branch chain amino acids, Wilson disease, low copper chelators, vitamins, some metabolic disorders respond to high dose vitamins, hormones, growth hormone deficiency, genetic engineering, gene therapy is experimental but promising for some disorders. Pop quiz. What is the term for the genetic trait characterized by extra finger or toes? A. Talipes. B. Polydactyly. C. Osteogenesis imperfecta. D. Neurofibromatosis. I will pause for a few seconds. Think about it. And the answer is B. Poly or polydactyly. What's the term for the extra finger or toes? That is polydactyly. Talipes is the clubfoot. Osteogenesis imperfecta is brittle bones. Neurofibromatosis is nerve tumors. So the answer is polydactyly. Role of genetic counselors. Genetic counselors are health professionals with specialized training in medical genetics and counseling. They work with physicians, nurses, and lab staff. They collect family history, interpret lab results, explain inheritance pattern, and help families understand risk and options. They do not make decisions for the family. They provide information so families can make informed choices. That's wrap up chapter 25. We covered genes, chromosome, alleles, dominance, meiosis, Punnett square, sex-linked traits, mutation types, congenital versus hereditary, specific genetic disorders, prenatal screening, and genetic counseling. For your exam, focus on the difference between genotype and phenotype, how to do a simple Punnett square, the inheritance pattern of X-linked recessive disorder, examples of autosomal recessive and dominant diseases, and the indication for genetic counseling. Thanks for watching. I am Dr. Maryam. See you in the next chapter.